Oh, it's a pleasure again. So we're going to continue on the theme of how cells move, maybe with a slightly different focus today from the perspective of the fluid, a little bit more rather than from the cell. So again, just a quick overview of the overall outcomes for these three lectures. I hope yesterday you gained some intuition about how organisms in general, microorganisms in general, manage to solve the fairly complex problem of swimming or moving at all at low Reynolds numbers. And then we identified some common themes, in particular the usage of these filamentous, these filaments for propulsion, and presented an argument for why this is important. And then what we want to do a bit more today is to really understand, connect these movement patterns with experimental data in order to understand how organisms interact with their fluid environment. And then hopefully tomorrow, armed with these dual perspectives, if you like, we can then try to ask how, specifically in different case studies, in different species, how these organisms can actually actively control their movement patterns, meaning that they can manipulate the activity of these diverse appendages for different functions. Okay, so again, a reminder, just so that we never see a confusion between the terms flagella and cilia again in the literature. And so what we mean by cilia are these thicker compound structures that are found only in eukaryotes. In fact, in most eukaryotes, you have a cilium. It's much thicker. It has many more, I like to think of many more degrees of freedom, thanks to many molecular motors that actively generate force all the way along this filament. And you might also hear that they're referred to as eukaryotic flagella, but it's not the same as a bacterial flagella or an archaeal flagella. And so maybe just a remark on archaea. So these tend to be more of a less well-known, let's say, kingdom, although that word is forbidden in some circles, that we don't think about as much as physicists. But nonetheless, they actually, of course, they're subject to the same physics as these other two types of organisms that we're more familiar with. And then, sorry, I'll talk about this first. And then if we wanted to do just a very simple comparison about between bacterial filaments, flagella filaments, and the flagella filaments that make up archaea, or archaella, as it's sometimes known, they are very similar. So the filaments themselves all comprise of intrinsically helical subunits that somehow arrange into these compound superstructures that have some sort of predefined or well-defined helicity and parameters. And what is really the distinction is in the way that these filaments are assembled. And rather interestingly, and I think more people, especially biophysicists, should maybe study this process. And the distinction is that unlike bacteria filaments or flagella, which are assembled from the tip to the base, archaea are actually assembled in the opposite direction. Cilia, on the other hand, are also assembled from tip to the base. And so, as I was talking about yesterday extensively, the purpose of having a slender filament is that this allows you to exploit something known as drag anisotropy. So basically, in the limits of something like a spheroid, where you extend the aspect ratio between the length and the radius, if you like, eventually you get this sort of scaling where the drag coefficient in the perpendicular direction is approximately twice that in the parallel direction. And then one thing to note is that this ratio can never be exceeded, so you can't really get better than this. And so I just wanted to sort of highlight that in nature, this exploitation has been done to a remarkable extent, in the sense that not only do you have filaments of the types that I already described, different species have also come up with, let's say, filaments upon filaments. So what is this? This is a single-celled organism called Ochromonus, which has two of the cilia or eukaryotic flagella. One of these is shorter than the other, and we believe that only one is actually used for propulsion. We can discuss what the other one is for another time. And intriguingly, this is an artist's impression of some electron microscopy data, where you can really distinguish all these very interesting sort of partitions in the main structure, main cilium. So these are called mastigonemes, and they come in all sorts of flavors. You might think of them as merely extensions to the cilium itself. So imagine that you embellish the rod now with many rods aligned perpendicular to the main axes of the cilium. So unlike the cilium itself, these mastigonemes don't have motility intrinsically. So it's not that these filaments are able to move. They're just passively carried along. They're features of the morphology of this structure. And so if you imagine, and I'm not even sure if a rigorous argument exists, but just intuitively, what's happening here is that you're modifying these drag coefficients that I mentioned previously, we discussed yesterday. So we basically modified the drag coefficients and the balance of the drag coefficients in the two directions by the presence of these perpendicularly oriented accessory filaments. And of course, for this to work, there are some conditions. For example, these filaments really have to be oriented perpendicularly to the primary axoneme. But whether or not, so this really led to quite a lot of controversy in the field about whether or not the addition or the presence of mastigonemes on cilia actually helps propulsion or not. But what's undeniable, and I wish I had a video of this because we have these organisms in the lab, what's undeniable is that in this case, when you do have rigid filaments oriented in this way, the organism propels itself forwards. So instead of, in the case of a naked filament, if you propagate away from left to right, the organism gets propelled to the left. But in this case, if you watch a video of this organism swims to the right. So why should you do this? I mean, this is not a question that I will answer, but I think it's useful sometimes, or in fact, all the time, to ask yourself in these biological systems, why do you find a certain phenomenon? And is it there purely because of some evolutionary accident, or is it a constraint of the physical environment? Okay, and because Margaret is in the audience, this is one of the first papers that was published in PRX Life. So this is an interesting study on the effects of mastigonemes, a computational study. Not so much, well, propulsion as well, but also other functions of these cilia include feeding. So life at these scales, despite the fact that obviously in these lectures, I'm focusing primarily on swimming processes, organisms at these scales also use the same appendages often for feeding instead. And if you can imagine, you're just about 10 times the size of a bacterium, that already allows you or gives you a chance to feed on these bacteria, or in fact on nutrients in the solution. And so, but you don't often have, let's say, the luxury of creating different purpose-built appendages. And so what you do instead is just use the same appendage that you normally use for swimming, for feeding instead. And this is kind of what I wanted to emphasize in terms of active actuation mechanisms, because this is something that I like to think of as being more accessible to eukaryotes than prokaryotes. Because if you think about prokaryotic flagella, these are rigid helices that can only be actuated from the boundary. And therefore, there isn't the space or the class of solutions that you could change are really limited. So in this case, it turns out, as you might expect, having this very fluffy or decorated flagellum enables an enhancement in feeding. And this may be important in some certain types of organisms. Okay, so what about, this is just a sort of highlight about the importance of the intrinsic properties of these filaments. So clearly, evolution has, to some extent, optimized these properties to make them more efficient at propulsion or more efficient at feeding. And it's useful just to think about concepts that you may have already encountered over the course of the last few weeks. So one of these is this idea of a persistence length of filaments, which you can just consider as the ratio of EI, this bending rigidity, to thermal fluctuations. And this quantity, this ratio, has dimensions of a length. And you can think of the physical interpretation as at which point or how long does it take before these different segments decorrelate from each other. And so if you are a filament that is much greater than your intrinsic persistence length, then under thermal fluctuations, you'd expect it to wave around and wriggle around. And in that sense, it would become unable to maintain the preferred shape, which, as we know, is essential for swimming. Of course, you may also encounter the other limit, where it may be too stiff. And then, again, thinking about cilia, there's no way then you'll be able to modify the bead pattern. So these are just some typical values that you might encounter the persistence length. And so from this, or you can appreciate that the persistence length of these filaments is also directly related to this bending rigidity, which is a property that is very difficult to access experimentally. And in the literature, and yet it's such a critical parameter. So if you're intent on creating computational models of these structures or microswimmers, then this is an important parameter that you have to input into the simulations. And so often there are only a few sources that are quoted. So for bacterial flagella, I'm not sure how much I really believe these values. These are the values that are quoted in the literature, but always really take these as with a pinch of salt. And then I group these according to prokaryotic versus eukaryotic appendages. And what you find, I think the order of magnitude is probably correct. So typically for motile cilia, this is on the order of 10 to the minus 22 newtons meters squared. Whereas for primary cilia, so these are slightly strange objects that are found pretty much on every single cell in your body. These don't have motility function, but may have rather sensory functions. I won't talk too much about that. But these have bending rigidity that's around an order of magnitude less. Okay, so how did people go about measuring this parameter? And so there's a variety of ways. I would say that this can be summarized into either subjecting these filaments to flows, or subjecting them to some sort of controlled load. So for example, in this case, this is a cell. This is Chlamydomonas, which you may have, well, I mentioned this yesterday. So this is a single-celled organism that has these two cilia, which one of these is in focus, the other one is slightly out of focus. And obviously, as I mentioned yesterday, if these were beating, the cell would be moving forwards and backwards, forwards and backwards. But if you were to resist the beating, firstly by holding the cell, the flagella or the cilia will still continue to beat in the same way, pretty much. And then if you were then to come with an optical tweezers, this is a small microsphere held in an optical trap, and then gently manipulate this filament position with a known calibrated force, then you can use sort of beam theory to then reverse calculate the bending rigidity of this filament. Is that the best way? I'm not really at the moment. This is the best approach that exists, but if you're interested in exploring alternatives, please let me know. So one thing that in the history of the subject was quite a critical observation was the idea of wave propagation along elastic filaments. So elasticity, it turns out, brings something different to the story. So this in this very seminal study, this was actually an attempt to understand how cilia can actually propagate bending waves. So if you imagine a fairly long cilium, for example, a sperm flagellum, you might imagine that the wave, or at least from the calculation we did yesterday, the wave retains its amplitude as a function of the length. But is this actually possible to generate with something known as boundary actuation alone? So if you imagine sticking, say, a cilium to the apparatus that is used to propel bacterial flagella, is that even feasible? And so in this case, we imagine a balance between elastic and viscous moments. And this is directly taken from the original study. And you end up with a PDE that looks something like this. I'll explain a bit more in the next slide, subject to these boundary conditions. So what are these boundary conditions? So imagine that you're wriggling an elastic filament from the end, then at the x equals 0 end, obviously y is 0, and then the movement is described or can be described by an oscillation. So if you are wriggling it with some set frequency, let's say. And then at the free end, you have torque-free and force-free boundary conditions. And this you can solve. And what's interesting is that the solution revealed something fundamental about the mechanism of operation of these cilia. So just to go into this in a bit more detail. So I think we, maybe in the previous lectures, have encountered something like the definition of a bending energy for structures, elastic structures. And then if you basically consider a filament and you apply a bent filament will naturally be associated with an elastic force. And this force can be found by differentiating the elastic moment, the associated elastic moment. And this critical parameter, this bending rigidity, once again appears in these equations. So you might recall from yesterday, the transverse force, transverse hydrodynamic drag, which I do not have on this slide, is given by this term on the left-hand side here. So if you recall the derivation that we did yesterday with the planar undulatory wave, you might remember that in the transverse direction, you have this zeta xi perpendicular multiplied by these deformations in the transverse direction. And so if you set a balance between these elastic and viscous forces, you get an expression that looks like this. If you ignore these parameters, then the general form of this PDE is something known as a hyperdiffusion equation, which admits certain well-known solutions with some properties. And what's particularly interesting is if you then considered activity. So instead of having a passive elastic filament, now you added, imagine that this is the interior of one of these cilia. Imagine there are many molecular motors operating on these filaments. Then activity is going to lead to to a third contribution. And the reality is that inside the axoneme, you have this extremely complicated ultrastructure, which unfortunately I do not have a picture of this. But if you imagine taking a cross-section through the cilium, there's actually this very characteristic and very well conserved arrangement of microtubule doublets. Maybe I should try and, I'm not going to draw all of them because there are 9 plus. Is there a chalk? Okay, I'll just take the yellow one. So this is an attempt, axoneme cross-section. So imagine that picture where you're taking a cross-section through it. So this is something in the center known as a central pair, which in some organisms actually mysteriously rotates as the organism propels, as the waves propagate. Okay, imagine there are 9 of these. So this is a microtubule doublet. And molecular motors, known as dynein, basically walk along these filaments. So imagine this is walking into the page. So the reality is that you have this extremely complicated ultrastructure that to this day we don't really understand. Do you have a question? Yes.
What's the distinction between axoneme and cilia?
Well, it's just a matter of terminology. So I think it's just you would have something known as a ciliary axoneme to describe this type of axoneme. But there may be, I think it's a fairly specialized term that you find mostly in this context. But equally, if you were to manufacture one of these using synthetic mechanisms, you can still call that a synthetic axoneme. It's just a fairly specialized term. And so to date, we still don't understand how this thing operates. But in a very sort of simplistic picture, you can imagine that instead of having the full 3-dimensional ultrastructure, you just reduce the whole thing down to two filaments, two parallel filaments, where the forces are generated by molecular motors walking along these filaments. So that's why these forces in this diagram are pictured oriented in tangentially to the filament. And then what happens is that you would have, because of the bending, some amount of shear. This shear may be non-zero at the base, but in particular, in general, you'd have to have this expression, which accounts for the separation between these two parallel filaments and the arc length dependent curvature along the filament. And so with this type of simplification, many authors, including this reference here, have attempted to really understand or modify, come up with modifications to this sort of basic force balance that is associated with passive filaments in the case of these highly active structures. And this is, again, a rich literature where full of controversy about the origins and the coordination of the activity inside these structures, because you can imagine that all of these simplifications are really very poor representations of what actually happened in the real filament. And all of these models, to some extent, will capture under some regimes. You can start to, it becomes very complicated very quickly. So in certain regimes, you can get patterns, beat patterns. So beat patterns meaning solutions to this equation that resemble what you find in real cilia. But does that really mean this is the underlying mechanism? I don't think you can quite make that connection. For example, there's a class of models where you don't have a slide for this, but there's also a class of models where you imagine you have this filament which is mostly passive, except for some sort of a follower known as a follower force. This really should be directed tangentially along the end, the tip of the hypothetical filament. And surprisingly, at least to me, this type of configuration, so this is known as a follower force model. This type of very simple configuration actually admits oscillatory solutions. And there are some studies which suggest that just because of this correlation, maybe this is a mechanism for ciliary actuation. But this is again an instance of does correlation really imply causation? Okay, so I hope that sort of summarizes essentially the factors that are always in play whenever you think about how these elastic filaments are actuated. So going back to Machen's original analysis, why was it so compelling back in the 1950s? It was this very simple realization that essentially the amplitude that you get along the filament has to decay exponentially along its length. So compare this to what you find in experimental observations. So this is something that one of my PhD students took. This is not a sperm, it's just a different organism that will remain nameless. So this is a sperm-like cell that actuates a cilium. So you can really appreciate the bending waves that are propagated along this object. So we've kind of relocated each of the frames so that it's this appears to be stationary. And these amplitudes really don't decay as predicted in Machen's original study. Yes? The first one? Yeah. This is inextensible. I would say these filaments are inextensible. I think it's something that you can measure experimentally. So on the whole, they are. This is assumed. But you're right. I mean, I'm trying to think of a. I can't immediately think of a scenario that would. But let me think about it a bit more. Maybe there's some situation where this is more important. But this is common. Doesn't matter anyway. Okay, so that's another way to look at this. Very good. Yes, so going back to this analysis by Machen. So remember this was back in the 1950s, where there was no technology to image the interior of these ciliary axonemes. So nobody at that point knew about all these structures, and so this was like a really important finding that if you were just to wiggle a passive filament of the correct length and material properties, this is never going to get you propulsion or the dynamics that are observed in real organisms. And so because of this contradiction, it means that there must be active forces all the way along these filaments and not just at the boundary. So this is a fundamental distinction between eukaryotic appendages and prokaryotic appendages. Okay, and what's interesting or particularly useful is again, if we go back to these equations here, this one in particular, this is associated with a scaling or a balance where you can construct another quantity known as the sperm number, which is a ratio of two length scales, so the length of the filament in question, and this LP, which is if you look at the moment, the balance here is somehow this very interesting ratio. And for different organisms, for sperm, real sperm, this number is much larger than for something like a Chlamydomonas cilium. And this really characterizes the dominance of viscous versus inertial, elastic forces. Yes?
What are the boundary conditions? Is it fixed and free?
It's fixed and you're oscillating. That's the, well, in this particular case. Okay. Right. So this is after all of this, we now obviously have tools to really visualize these structures in much more detail using techniques such as cryo-electron microscopy or cryo-electron tomography, where many authors and many groups have figured out the exact identity of these motors along the filament, and this actually falls into many distinct species that we think are important for regulating different aspects of the beat pattern. So for example, we think that there are some motors that are critical for regulating beat frequency, whereas other motors are important for regulating beat amplitude. And so together, somehow, in this three-dimensional architecture, they're able to generate this very dynamic beat pattern that we find. Okay, so what's interesting is that if you were to think about, in a synthetic context, creating some of these elements from man-made materials, creating a synthetic cilium is actually very difficult, precisely for the reason that you can't, it's very difficult to get motors that are small enough to fit inside this micro scale structure. And also on top of that, be able to control what these individual motors are actually doing at any given time. And so, unlike in the case of artificial bacteria-like micro-robots, where you just simply manufacture a helix, you attach it to some rotor and a head, and the thing will swim, it's much more difficult to do the same thing for biomimetic cilia. So one thing that we and collaborators worked on recently was a slight modification of Machen's original argument. So in this case, I've grouped all the other terms here, just have a single parameter for simplicity. And one thing that we did in the past was always assume that the bending rigidity, this key material property that I mentioned before, was always constant along the filament. But this really doesn't have to be the case. So you can imagine much more complicated scenarios where maybe the bending rigidity varies as a function of the length. And there are some studies, in fact, that have looked at the effect of the bending rigidity that does vary as a function of length. But maybe more interestingly, from a design perspective, what happens if the bending rigidity depends on the sign of the curvature, of the local curvature? So basically what this means is that imagine if you construct a filament that tends to bend or bends more easily in one direction than in the opposite direction. You would have this E, this bending rigidity dependent function that now depends on the local curvature instead. So we, in this case, revisited Machen's argument with exactly the same boundary conditions as before, but now applying the so-called state-dependent bending stiffness, which is different whether you bend the filament upwards or downwards. And so then it turns out that this very simple modification actually allows you to sustain much higher amplitude waves compared to the case where the bending rigidities in the two directions are identical. And so what this means is that we can then try to engineer a filament that looks like a cilium using boundary actuation. So this is, of course, not how real cilia operate, but if you're thinking about synthetic analogs, if you want something that has the same pattern as a biological microswimmer that uses cilia, you can simply break the time reversal symmetry by adding a series of hinges, or at least one hinge. And the effect of the hinge is then that you create this asymmetry in the bending stiffness. And so with this very simple modification, this filament then starts to execute time irreversible waves. And this allows, so the question is then, does this thing swim? If you put it in a fluid, in a viscous fluid. And interestingly, this story actually, our idea for this project came after this second slide I'm going to show you now, which concerns, this is a slight detour, but this concerns the question of how does a cell that has a certain number of these appendages change gait, or what is the effect of gait on its swimming pattern? So to understand this, we first look at the biological context, where it turns out you can find many different species of these micro swimmers that look broadly the same. They have the same body size, mostly, and the same number of appendages, in this case 4, but they have different gaits of swimming. So this is obviously a projection microscopy video where you can see them alternating between this trotting gait, where you have one pair of breaststroke in one plane and then another pair in the transverse plane. And then you have this simultaneous gait where all these appendages are actuating at the same time in phase with each other. And then you have this galloping gait where you have this strange rotary gait. And so our question originally was, does gait in this context make any difference to how much you swim, let's say, as a function of one beat cycle? So individually, going back to yesterday's lecture, individually, each of these cilia are doing approximately the same stroke pattern. But then if you have 4 of them, and then you're actuating them in a different sequence, does that actually matter? So in this biological context, we just set them against each other and measured how far does the cell swim as a function of the gait. So unsurprisingly, in the trotting gait, you're just making net forward progress all the time. And then this changes depending on whether you have other variations. So our original idea was to try and create a synthetic version of this type of cell that would allow us to really reproduce this type of plot. And so this was where the idea for constructing a macroscopic synthetic microswimmer first arose. So the thing that we had to emphasize was, of course, if you want low Reynolds number movement or physics, then you need to recreate the same environment. So because of design limitations, it's very, as I mentioned, very difficult to miniaturize these things and create motors that are small enough, let's say microscopic scale motors. So it's much easier to work with an upscaled robot and then change the viscosity. And so for this particular experiment, in collaboration with Dan Goldman, what we did was place one of these artificial robots inside a large tank of glycerol or mineral oil. So this obviously increases the viscosity many-fold, and then we can calculate the Reynolds number again, and assuming it is very small, although not as small as what the organisms would typically experience, you can start to ask this question. So with a very poorly constructed gate and a poorly constructed waveform, does changing the gate actually influence the swimming dynamics? So this is what this robot looks like in dry air, and then you place it in the fluid. This is just to convince yourselves that this can reproduce different types of gates. So you can basically first test whether low Reynolds number hydrodynamics makes sense in the situation. So if instead you were trying to propel with a completely rigid rod that does not have the hinge, so it doesn't break symmetry, the front and the back strokes are identical, does this thing swim? Not, thankfully not. But okay, we didn't bargain for the slight sinking that you may have spotted. But if you ignore the sinking, it's not actually making forward progress. So in this context, this is consistent with observations that for a macroscopic realization of a microswimmer placed in this high viscosity environment, is not actually able to swim with a rigid stroke. And then, of course, if you start to increase the stroke frequency, So this means that you're going further and further away from the low Reynolds number regime, then you do start to get net progress and inertial effects. So there's only so much we can do here, but the point I wanted to make after all of this is that we can then use this to it's the question, does gait matter? So if you change the gait in this, let's say, firstly biflagellate robot, so watch the center, it does really swim. If you change the gait, this is a side view of a quadriflagellate version. You can appreciate that you can start to make the same measurements that we did for the real biological microswimmers, and again measure or quantify the effect of this gait or the effectiveness of this gait as a function of a characteristic beat cycle. So how many body lengths does this macroscopic robot move per cycle? And you can start to reproduce exactly the same profile that we found in the real organisms. For example, the trotting gait is the best in this scenario. So this is a little bit of a detour, but I hope it's useful to think about in the context of manufacturing devices. Yes?
So if the biological context, if the trotting gait is so good, why do you think they ever do?
Yes, that's a very good question. Again, this is where why questions are generally more difficult to answer than, let's say, how questions. I mean, I have some ideas. We can also discuss this more, but again, it goes back to the idea that in nature, you're not just trying to swim faster. This is not your primary or only objective, because these cells, the objective of these cells is really to multiply, grow, and divide and propagate the species. And so maybe there are situations where feeding becomes more important. I'm not saying that these particular organisms that we study actually feed in that sense, but I think it's one of these situations where it's not just a question of how fast can you swim. And there are other constraints as well in terms of the cellular architecture, which I hope to mention tomorrow as well. Okay. Okay. So the summary for this first point so far is that you have to break symmetry with these appendages. So in the case of bacterial flagella, you break the symmetry by actuating a chiral object, such as a helical flagellum. In the case of an undulatory swimmer, like a sperm, you propagate a wave, which breaks symmetry. In the case of these shorter appendages, like cilia, you break symmetry by breaking the forward and backward stroke patterns. Whether you do that with a hinge in the case of the robot, or in the case of the real organism, well, this is still a little bit mysterious. If you go back to thinking about the ciliary axoneme, how do these different motors then conspire to change the pattern of the forward versus delivery strokes? So one thing that is a little bit of an interesting aside is that you can still achieve net propulsion, or rather, if you were to fix the cilium, net pumping of fluid by actuating a completely rigid filament that has no front-back asymmetry. So the way that this happens is by using boundaries, or the influence of boundaries in the environment. Situated in a completely free environment, then you're not going to get any net propulsion. But in the case of this is the vertebrate node. So this is a really fascinating situation where these appendages play a really critical role again, not so much for swimming, but it's a very different function. So it's known that in many species of vertebrates, symmetry breaking occurs in following specific sequences. So you would, I think, break the dorsal ventral, create the dorsal ventral axis first, and then you would have the anterior posterior, and then finally the left-right symmetry axis is the last one to be broken. And so it turns out that cilia are critical for this process. And so there are many studies that have tried to understand how this process happens when you have what appears to be a fairly randomized distribution of these individual, they're called nodal cilia, whirling around. How do they actually collectively create directional flow in this region known as the node? But I think for our purposes, we just need to note that the presence of the solid boundary has a really critical effect on the effect of the environment, on the flow structure around one of these appendages. And so to understand this a little bit more, this brings us to the notion of multiple expansions. So this is also has been set as an exercise. So I'm not going to go too much into detail here, but the critical observation is that because of the linearity of the Stokes equations, what you have is that the derivative of these solutions are themselves also solutions of the same equations. And therefore, if you take the Stokelet that we discussed yesterday and differentiate it, and then differentiate it many times, you end up with these higher order solutions, which maybe some of you are exploring in the exercises. So this sort of table really just summarizes some of the leading order contributions. So if you take these arrows are really supposed to represent the physical interpretations of these derivatives. So if you take a Stokelet, which is represented by, again, this forcing at some position, and then if you take a pair of them oriented, let's say on a line collinearly with each other, you get something that is known as a stress. And then these obviously don't have to be aligned, and when they're not aligned, you might end up with these two contributions. This is kind of giving away the solution to one of the exercises. You end up with these two contributions, one of which is the stresslet above, and the other one is a different kind of flow pattern that's associated with a point torque in the fluid, which is known as a rodlet, but also sometimes known as a couplet, depending on the age of the literature. And then there's a second class of solutions. I will need to type this up. There's a second class of solutions associated with, they're similar to sort of what you get in potential flows. And for that reason, they're known as potential dipoles or potential quadrupoles or even higher order solutions. So again, this comes from the fact that you can repeatedly differentiate these flow fields to get flow fields that themselves satisfy Stokes equations. And then another brilliant property of linearity is that then you can sum all these solutions up in some arbitrary order, and then you would still, the sum would still satisfy Stokes equations. And so together, on the slide, basically, you find all of the important contributions to understanding the basic structure of flows generated at low Reynolds numbers. And what's always important to keep track of is the the order of the solution. So in the case of a stowstud, this really just scales as 1 over the distance from the source, from the stowstud, whereas the leading order contribution in the case of the source is 1 over r squared. And the Strausslet also scales with 1 over r squared. And the implications of these scalings will become important again in as we delve into how this relates to real data around microswimmers. Yes. Hi. Yeah. Which one? You mean this one? It should be x to the 5th, I believe.
The overall power.
Overall power is 4, but there are x's. So just in, I think this is correct.
But like a first term, like 1 out of 15 in front of it, is it 3 over 3?
That's 3, yes. Yeah, good question. Anyone know these solutions by heart? Okay, I should check this then.
More physics.
Yes.
Like on the previous slide, you talked about, yeah, like
this one. Yeah.
There must be some quantitative, measure of how non-reciprocal and where you get first stroke.
Yep. Yeah. This is. No, I wasn't planning to. I wasn't planning to discuss that in detail. Did you have a? Is it understood? I think the way to quantify it is well established. So, for example, you can construct say symmetry parameters, or rather measures of asymmetry, which take into account not just the shape, but also the time that you spent doing the power stroke or doing the recovery stroke. And so to that extent, the quantification of any experimentally observed pattern is understood. But are you asking how are these generated, or is that?
No, how generated, but like to quantify, like you give me a pattern that
Yeah.
And there's some number I can calculate to say how non-reciprocal it is and how much propulsion per stroke that would
get. That you can do. And in fact, this, how do I answer that? So I think in the case of a single cilium, this is well understood. In fact, the paper that this stroke pattern is quite a famous stroke pattern because people have, for computational simplicity, you use this pattern as the basis for calculating what happens when you have arrays of these types of filaments, or you arrange them in different architectures. Using just like a Fourier expansion tells you, it simplifies the computational architecture. But I think, yeah, the case of multiple filaments becomes much more complicated. But yeah, that wasn't something I was going to mention more. Okay, I'll check that and definitely typeset it. So, okay, so coming back to the real world context, so then how do we measure or match some of these flow solutions with what is actually observed around real swimmers? So this is just, again, the sperm-like propelling cell that I showed you before. So what we can do is this technique called particle image velocimetry, which is really very well established and used a lot in fluid dynamics to characterize the patterns of flow fields. And here, where basically the color represents the local speed of the flow, and then the streamlines are represented in white. So you can see that this is already a very complex flow pattern. So if you wanted to know what is the underlying summation of these higher order solutions that are required to generate this pattern, then this is a possible research question. And so just a quick slide about how this method actually works. What you do is really either in a large scale context, when you have a solution, a large volume of fluid, what you can do is create a laser sheet, which illuminates just one slice in this volume. But if you're in a microscope, then maybe you don't have access to a laser sheet, but nonetheless, just the nature of how a microscope works allows you to focus primarily in a very thin layer of fluid. So in either situation, you end up with a sequence or a movie consisting of a series of images that contain the positions of particles that you seed in the fluid. And obviously the particles will, don't know if you can see them very well in this video. So the particles will then move as the flow is generated. And so if you then take the image associated with one moment in time versus a later point in time, you can just construct or calculate this correlation between these two images as illustrated here. And these images are in reality, you can think of them as sort of surfaces in 3D, because at every point, you have essentially an intensity value at every spatial position in X, Y. And then if you take the correlation, wherever this correlation is highest, this means that this, let's say, region in the first image must have moved to this equivalent region in the second image. So then that movement is then associated with the velocity, and so this is hence the name particle image velocimetry. And so there are many software tools that exist that will allow you to then compute these things for your experimental videos. So, okay, going back to these fundamental solutions we just discussed, it turns out that if you think about what I said about swimmers, you can't really have net force, which is what a Stokeslet is. So there's no way that a freely swimming organism will generate, will have a Stokeslet contribution, except in certain cases that I'll come to. And so in general, you'd expect the flow fields around these swimmers to have a sort of stress-like profile. And generally, these can be further classified into either pushers or pullers. So in the case of pushers, the picture that you should have in mind is that there are two Stokeslets aligned with each other collinearly, and they're pointing away from each other. Whereas if you then flip the directions of these arrows, you get something known as a puller instead. So these drawings are supposed to represent, to leading order, the sort of flow patterns that are generated when you have a pusher profile versus a puller profile. Yes? No, I wouldn't say so. For example, here you see the arrows go away from the cells. Oh, right. Okay. Yeah. So this picture is somewhat misleading. What you find in in for example here is that there are some. It's not exactly what I've drawn. There are some subtleties in the near field. Yes.
I didn't know before that the flow fields look like this. And so I was wondering if interacting bacteria would, I mean, it seems to me that the interacting bacteria would have sort of pneumatic alignment so that they're perpendicular. And I was wondering if this is like a relevant enough effect and people like look at it.
Yes, they have. I don't know if this was discussed much in previous lectures, but this is something that people have looked into. So swimmers, depending, so exactly the profile around a particular swimmer will basically dictate how they interact hydrodynamically. So this leads to either alignment in the case of pushers or in the case of pullers, the opposite. But the thing is, if you're asking about the relevance in a more ecological context, is that okay? You're not asking about the ecological context. Okay, sorry. Yeah. Yes, I think I have a slide about this. Hold this thought. Yeah. They're not, in what sense are they opposites? Oh, yeah. So the dipoles are, yes, obviously. Yeah. No. What, is there a good explanation for that? I mean, in terms of the swim, yeah. I mean, it's true that so, I mean, I'm not drawing at all like what's actually happening in the near field. So I think this is related to the question that was it you asked earlier, actually. So I suspect it's because of differences in the near field.
I mean, this is symmetry that's got we just look at
Yeah. That's true. Yes. Yeah. But I guess if you were to construct, maybe in the case of colloidal swimmers or something, where they are actually spherical. Yes. Yeah. That's a good point. Sorry, can you just repeat the last thing you said? I don't think it's... I mean, if it's not a sphere, then definitely not the same.
It's like true that now I always confuse it because I use a different language.
Oh.
Pushers are unstable.
Right. Yes. But is that because we're assuming, but in those, is that with spheres? No, that's with, in the simulations, you mean? Okay. Right, there are probably some subtleties that we should discuss that I would be interested in discussing actually in a bit more detail later. But I think it's definitely the case that in most biological swimmers, they are never going to be exactly described by one or the other type of flow field. But they can come quite close, especially in the far field. And so you might have, so again, using this particle image field symmetry technique that I mentioned before, it's actually possible to derive or measure the flow patterns generated by different types of swimmers. So it turns out that your canonical biological version of a pusher swimmer is then an E. coli, let's say a bacterium, which, if you imagine what the effect of the rotation of the rigid helix, it generates a flow which pushes out behind the cell, and then the effect of the swimming is then that there is a flow which pushes out in front of the cell. And this leads to this sort of extensile regime where you have the dipoles oriented away from each other, roughly aligned with the axes of the cell, of this highly asymmetric cell. In contrast, if you have something like a chlamydomonas cell, which in this case is just actually was represented by a sphere, that's you're able to observe really the body dimensions in experimental images, and not so much actually the flagella shapes. But again, you can see that it's the opposite. So you have this interesting stagnation point actually in front of the cell, which is not captured by this very basic picture. And yes, I think there was a question earlier about whether you can change from being a pusher to a puller, and actually you can without doing very much. So one thing to observe or note is that in these flow fields that were measured in this article, these are time averaged flow patterns. So you can, if you imagine the PIV data, you have a frame for every time, and then you're just like averaging over all of these frames to get assuming that you have this fairly steady dynamics, steady but periodic dynamics. But if you then zoom in time, if you like, at what's actually happening during a beat cycle, so then it's not very surprising if you think about it, but it turns out that something like a chlamydomonas will actually oscillate between a pusher and a puller at different points during its cycle. But on average, what you get is this sort of polar profile that we tend to associate with chlamydomonas cells. So this is simulation results, not from experiments. I'm not actually sure if they've been measured. I don't think they've been measured, surprisingly, in experiments for chlamydomonas. Again, the emphasis here is on the behavior in the far field and whether it resembles this dipole flows. Yes. Is there a velocity difference in terms of, yes. In reality, well, it's very hard to compare. And you would have to, again, probably the best bet would be to create some sort of synthetic swimmer that has identical geometries. I'm not sure if that's been done. Does anyone work on synthetic artificial swimmers? Colloidal swimmers? No. Okay. So that question is open. Okay, so I mentioned that sometimes there are some situations where you can allow for the far field flow around the swimmer to be of a Stokes-Lit sort of profile after all. And this includes situations where you're applying some sort of external force or an external torque. So in this case, what we can do is hold a flagellated swimmer, let's say, fixed so that it's unable to move. And because of this, you can actually imagine that it's propagating as a wave down the cilia. Or in this case, this is just a cilium, a short cilium-like stroke pattern, which is just a power and then recovery stroke cycle. And this really does generate Stokes-Lit, a very fairly good match to a Stokes-Lit profile. Again, this picture here is what you get on average, the time average flow. And what's happening during a beat cycle is much more complex, let's say. But nonetheless, you can still associate at every time a Stokes-Lit profile for these types of flows. So what we're showing here is how the fitted Stokes-Lit, if you like, rotates during a prospective beat cycle. And so the picture that you might have is that it's actually possible to approximate the complex, the flows induced by the complex beat pattern of a cilium by that of a single Stokes-Lit that's moving around on an orbit in a time periodic fashion. And so just to convince you that this really is a Stokes-Lit decay, what you have to do is measure far enough away from the source, from the origin. How does the average flow magnitude vary as a function of distance? And so you'd expect the leading order behavior to be 1 over r, which is represented by this linear fitting. And the experimental data points really do lie on top of this. So to leading order, sufficiently far away from the swimmer itself or the source of the perturbation itself, the picture is quite well understood from a fluids perspective. And then the other situation you might think of where the picture, again, you're able to associate a Stokes-Lit term to the far field is when you have a large enough, right, which actually sediments by gravity. And so in this case, what's pictured here, again, it's in the same study, is this fairly large colonial alga called volvox. And this is typically on the order of 0.5 millimeter in size. And the reason for the reason for the sedimentation really, if you picture, this is meant to be is this uneven distribution of cells on the interior of this multicellular organism. So this guy has 2 distinct cell types. One are the somatic cells, which have cilia. So each of these smaller somatic cells has 2 cilia, which are used for repulsion. And then each of these larger cells in the interior, these are germ cells that are responsible for, basically, they become the next generation of volvox. So because of this uneven distribution, this thing will sediment due to gravity. And therefore, you have an external force, which makes a difference in this scenario. And then, as I mentioned in the previous slide, what you're looking for is to, if you wanted to match some expansion, multiple expansion to the data that you acquired, what you can do is then write it in this very suggestive form, where you have the first term is the uniform flow, background flow, because of the swimming, and then you have the Stokes-Lit term, and then the Stress-Lit term, and then higher order terms, which you can quantify. So in this picture here is what happens when you subtract the leading order Stokes-Lit term. And then what you get is this picture where, again, if you do the same thing that we did previously, which is measure how does the speed change as a function of distance from the center, you can see that by adding more and more terms, you start to match the data increasingly well. And so again, this emphasizes that for any given data set, it's actually, the picture in the far field is actually well understood. But if you then ask yourselves, okay, you have all of these thousands of cilia on the surface, deforming this, the local flow, how do they actually, what is the flow profile close to the boundary? So that becomes a much more difficult question to address. Okay, so the other consideration is, as I mentioned before, in the context of nodal cilia, is the effect of boundaries in the environment. And so this requires a modification to the original Stokes solutions, sorry, the Stokes-Lit solutions I mentioned earlier. And this expression, particular expression, is due to Blake, I think. And essentially, sometimes it's also referred to as a Blakelet, but it's not a universally adopted term. But nonetheless, it has very similar structure compared to the Stokes-Lit. You have these additional terms, and the basic idea for this is that if you have a solid boundary, you must still satisfy the no-slip condition at the boundary. And the way to do that is very, where this is quite an ingenious method, is to imagine that you have an image solution on the other side, and then if you sum these up, this allows you to then satisfy no slip at the boundary. And in this case, you have additional terms that arise due to the position of these additional image multiples. And the way to interpret this is then, in this case, you have two contributions from, one from the distance to the original Stokes-Lit, and another contribution, or another set of contributions, due to the distance to the image instead. So this is really an ingenious invention, and I just wanted to maybe discuss the consequences of this for certain types of organisms. So if you wanted to really expand all of these terms, I mean, not sure if you want to do that, but in the case of this slightly simplified case where you just have a Stokes-Lit at the origin, sorry, you have the original Stokes-Lit is positioned, let's say you have a three-dimensional picture, a three-dimensional environment. It's in the origin in the plane, but at some height, let's say h, from the surface, the solid boundary. So if the Stokes-Lit were oriented so that it was perpendicular to the plane, then you can convince yourselves or do the exercise that this expression then becomes something like this. And this is the associated flow profile that this configuration would generate. And what this actually means for organisms is quite interesting, because in different species, you find, again, this is no longer about swimming, you find different configurations of these cilia, typically, that generate completely different flow profiles. These are typically used for feeding purposes. And in this case, this is no longer freely swimming, but these types of cells like or preferred the configuration that they often adopt is to sort of attach themselves to some sort of boundary where they then adjust the orientation of themselves, the body axes, according to whether well, who knows why they adjust the orientation of the body, but the consequences for the fluid physics is that then it generates different flow patterns, or even if you were to generate the same profile with the cilia, depending on the location of these solid boundaries, the effect of this is going to be very different. And so there are studies, for example, where people have considered the effectiveness of different profiles. Imagine different configurations of these stokes, different positions of these stokes relative to the solid boundary. Which of these profiles is more advantageous than others for the purposes of pumping fluid through itself, through the organism. And these are generally filter feeding organisms with the idea that that they have ways to filter the fluid. So imagine that the food or the source of nutrients is being carried along in the current. So there's obviously a drive to try and filter as much fluid as possible through yourself. So the more fluid you clear, then the higher potentially the chance you have of getting the nutrients that you need. So there are other, I don't really have time for this. There are other consequences of these flow structure interactions, if you like. One of them has already been mentioned earlier, and this is to do with alignment of multiple swimmers. And depending on whether you are a pusher type swimmer or a puller type swimmer, these alignment interactions will dictate the collective dynamics of these swimmer suspensions. And this is probably something that will be covered in, or has been covered in previous lectures as well. So I wanted to sort of end today with this just a fairly old study now, where we really try to investigate whether or not the effect of whether individual cilia interact through these hydrodynamic interactions through the fluid. And this is something that was first raised by, this possibility was first raised by G.I. Taylor, and again, with some ideas about why this might be a good idea. Why is it that having nearby cilia synchronized or phase locked in a way that resembles bundles of filaments, that really, what's the actual functional advantage of these types of configurations? And so this was had always been a hypothesis that maybe the theory would suggest if you have single cilium creating a perturbation in the fluid, this is going to affect neighboring cilia. That's kind of a very intuitive assumption, but is it possible to actually synchronize their movements through these types of interactions? So that remained unknown. And so we did the study, which I think is still relevant, especially for sort of pedagogical purposes. Here, what we did was we went back to this organism Bollocks that you might recall, and remember that each of these cells on the surface, that the somatic cells, they have cilia, and so it turns out you can actually remove the somatic cells entirely from the colony, and they will still continue to beat their cilia. So we can use this feature and to manipulate the relative spacing between nearby cilia and watch if they and how they synchronize or not. So obviously, as this video suggests, they really do synchronize their movements. And so this is, again, just a summary of what we did. And if we then, again, do this for many pairs of cells in different configurations with different inter-cell spacings, you can then get a sense of how the synchronization or the coordination changes as a function of the separation between these cells. So, for example, here, these are the same types of data that I showed you in the previous slide, except now I've removed the background, so you're left with only the two cilia that are oscillating or interacting with each other. So you can appreciate that when they're very, very close together, their movement patterns appear to be coordinated. As you start to move them further and further away from each other, this coordination starts to become less and less robust. So this can be then quantified rigorously in terms of sort of this phase difference. So imagine these are phase oscillators, two-phase oscillators. Then the relative synchronization can then be quantified in terms of the phase difference between these oscillators. So if the oscillators were perfectly coordinated or perfectly synchronized, then this phase difference should be 0. But if, as you start to move these oscillators further and further apart, they start to lose synchronization, then that phase difference should start to drift and grow without bound. And so we can measure this, and also, I won't go into too much detail of the experimental strategy, but we can measure this and then deduce a coupling strength that's associated with each data set. And so this coupling strength, you would then expect to decay as a function of separation, and with what scaling, and that really then depends on if you have, say, a Stokes step in a fluid, then the scaling should be 1 over distance, 1 over r, as we saw before. And so this is exactly the scaling that we find, and this really is evidence, I think, I won't go into this. This is really evidence that nearby cilia really can synchronize through hydrodynamic interactions in the way that we suspect. But that's not the full story, and that will be continued tomorrow. All right, thank you. Questions? Or you can ask me at the end. Other questions? Yep.
So in one of the beginning slides, it showed that the cilia has a series of, if I can remember correctly, those like rod-like particles.
Yeah.
So I'm just curious, what is the difference between those particles and like another kind of cilia which are aligned orthogonally to the central cilia?
I'm not sure if anyone has measured. Based on, as I discussed, the only ways to measure the stiffness is really by flow or by optical tweezers. And at some points, you just really don't have enough resolution, especially in sort of spatial resolution, to see the structures. But you can sort of make some predictions based on what's known about the material. And I think, then the arguments about whether or not these function to change the resistive coefficients or not really depends on whether they're rigid or not. So it is quite important, but we don't know a huge amount. Yes? Yeah. Yeah.
0 with some fixed phase difference.
Yeah.
If they're like infinitely far away, presumably they are not interacting, and so there's any phase difference stay at what it is.
Yeah.
So that's like going from like in if we consider like the dynamics of that phase difference, right? That's what I'm
Yeah.
invariant circle.
Yeah.
Big dynamic. Have you guys like quantified the bifurcation?
Yes, I don't have that slide here, but maybe I could show it next time. Not quite in such detail that you're visualizing, but we do have measurements which basically suggest the existence of a bifurcation length scale. So if you imagine, below a certain length, you would have phase locking, but above a certain length, it will just, you no longer have phase locking. So we managed to reproduce that kind of picture from data, but that's as far as we thought about that particular approach. But thanks for the question. All right, thank you.